Hi friends, good evening. How are you? Hope all are fine. Welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about pBR322. pBR322 was the first artificial cloning vector to be constructed in 1977. This cloning vector was developed by two scientists, namely Bolivar and Rodriguez. So the name pBR322 has been given. P stands for plasmid, B stands for Bolivar and R stands for Rodriguez. Bolivar and Rodriguez are the two scientists who developed this artificial cloning vector. pBR322 it contains 4361 base pair. This vector contains two selectable markers. One is gene BLA. Gene BLA encodes ampicillin resistance protein. Another selectable marker is gene TTA. Gene TTA encodes for tetracycline resistance protein. This is the ampicillin resistance gene and that is the tetracycline resistance gene. So the ampicillin resistance gene and tetracycline resistance gene it provide resistance to antibiotic ampicillin and tetracycline. Like other plasmid, pBR322 also contain an origin of replication. This origin of replication is derived from another plasmid known as PMB1. PMB1 is another plasmid. This vector also contains another gene known as ROP gene. This ROP gene encodes a restrictor of plasmid copy number. This ROP gene is responsible for restriction of the plasmid copy number. This cloning vector it contains restriction sites for 40 restriction enzymes. These are the restriction sites. These are the various restriction sites which are present in this cloning vector. Out of 40 restriction sites, 11 restriction sites are present within the ampicillin resistance gene, whereas 2 restriction sites, namely HINT3 and CLA-L, are present within the promoter of the tetracycline resistance gene. The ampicillin resistance gene it contains 6 restriction sites. 6 restriction sites are present within the ampicillin resistance gene. And the two selectable markers, that is ampicillin resistance gene and tetracycline resistance gene, are derived from two vectors. Tetracycline resistance gene is derived from a vector known as PSC101, whereas ampicillin resistance gene is derived from another recombinant plasmid known as RSF2124. This is about the artificial cloning vector pBR322. Hi friends, this is about pBR322. Please share, like and subscribe this video, and also don't forget to give your comments so that it will be easy for me to improve my performance, so that it will be more useful to all of you. Bye bye, good night.